Hello, everyone, and welcome to In Plain English, the podcast where we make science approachable, open source, and jargon-free. I'm your host, Jamie Moffa, and today we will be delving into part 3 of Interviews in Pain Research, featuring discussions that I had with researchers at the United States Association for the Study of Pain conference last month in Seattle, Washington. As always, you can find the posters discussed on this episode, as well as previous episodes of this series, on the website at inplainenglishpod.org. So without further ado, let's get to the science. And can you start by introducing yourself?
Hi, I'm Amy Nippert. I'm a researcher in the topic lab at Stanford University, and so I study basic mechanisms of chronic pain. I'm focused on the spinal cord, and I'm looking at a mouse model of a pain disorder called complex regional pain syndrome. And that's a disorder that occurs after an injury, a surgery, often a fracture and casting. And instead of healing up, like a lot of people break bones, most people heal up, thankfully. In a small subset, there's residual pain that has kind of a distinct acute phase where you have like red, hot swelling, and then a chronic phase where the limb is cool and atrophied, and you have pain in both of these phases. Mm-hmm. It's very hard to treat. So we're both using it as a model of sort of an acute and chronic stage of pain, and then also our final goal would be to find some therapeutic treatments. I'm focused on a cell type called astrocytes. So astrocytes are a type of central nervous system cell that's kind of canonically known to have a lot of housekeeping functions. So neurons are the cells that are sending signals to and from your periphery through your spinal cord into the brain. But the astrocytes are there to support those neurons to recycle things that they express or release into the environment. They're there to provide metabolic or nutritional support to the neurons. They provide structural support. And they also can participate in how much these neurons are signaling by how quickly they recycle neurotransmitters and other things like that. So even though they are mostly there as quote-unquote support cells, they also take an active role in shaping how many signals you're getting from the periphery to the brain. You can think about chronic pain as being a problem of too much signaling. A lot of times a light or normal touch will cause a really intense pain feeling, and that's an amplification problem, right? So I'm interested in looking at some of these sort of markers using RNA expression, which is a proxy for generally how much a cell is expressing of a different types of proteins. And I'm focused on ones that are important for homeostatic functions that astrocytes do. So these astrocyte housekeeping functions, are they changing in pain? And what you see is in the first stage of pain, right after a tibial fracture, you're actually getting a big upregulation. So you're getting a big increase in all of those markers. And those markers are probably trying to cope with the fact that there's a lot going on in the system, everything's kind of gone crazy. But we're mostly interested in that chronic phase. And what we see in that chronic phase is actually that they're down-regulating a lot of their homeostatic functions. So I kind of think about it almost as like if you just suddenly stopped cleaning your house or feeding yourself, that would be a problem. It's a problem in the system too. It may mean the neurons are acting in ways that aren't normal, in part because they're not getting the support they need from astrocytes. So we tried a couple interventions, including one where we delete some of the immune cells, or deplete, so we actually fully get rid of the immune cells in that area, and we actually find that improves the function of these astrocytes. So we're thinking that there might be some sort of interaction between that chronic inflammation and astrocyte function. And so our next goal is going to be experiments where we actually try to figure out how these two cell types are signaling, what signal is causing the astrocytes to downregulate some of their functions, and whether we can kind of get them to do their job again.
Right.
And improve, hopefully that'll be enough to at least improve the pain.
Right. So could there be like also beneficial immune cells in the area, or are they all kind of like contributing to inflammation?
Interestingly, in the CNS, you're really only getting one type of immune cell. It's an immune privileged organ, which means it doesn't really get a lot of immune signaling. So that main cell type is called microglia. They're similar to macrophages in the periphery, but they have a lot of specialized function. And we actually think that in the same cell, you're getting good beneficial and detrimental effects depending on the time post-injury and probably on a lot of other factors. So in a human model, we wouldn't necessarily be trying to just remove your entire immune system in the CNS. Instead, I think you'd be wanting to figure out how to maybe change their state to be a little less inflammatory, a little more resolution.
Right.
So there's a lot of research going on with microglia. I'm hoping someone figures that part out for me.
How do we shift them into that?
Yeah, so we did a big screen already where when we deplete them and they come back kind of, it's almost like turning the system off and on again. They actually come back like pretty fresh. They help improve pain. They almost look more like developmental microglia. And so we've done a large screen and found like which factors seem to be the most different. So those are going to be the first targets we go for. And hopefully through like, now it's the painstaking trying out all these different targets we found, and maybe one of them will be the one that kind of pushes them back into a more resolution state. And then that may also change their interaction with the astrocytes. So it's a really complicated system.
Yeah, no, it seems like it, but this is really cool. And thank you so much for going through it for the podcast.
Thanks for the interview. I will be sure to listen.
So can you start by introducing yourself?
Yes, I'm Matias Preisegger. I'm a postdoc in the University of Pittsburgh in the PCPR. I'm from Argentina, and I'm my third year postdoc.
Excellent. And then, so can you tell me a little bit about your research?
Yes, my poster is entitled Modeling Osteoarthritis Pain with a Tissue Chip. Osteoarthritis is a disease that affects the joint. Pain is the most prominent symptom why patients look for medical attention. So despite all this effort in looking for an osteoarthritis treatment, nowadays there is no osteoarthritis treatment widespread approved. So we suggest that this is due to the lack of a gold standard to study osteoarthritis and the limitations that the current animal models has. So our idea is to develop an organ-on-a-chip device to study osteoarthritis in vitro. To achieve that, we generate a cartilage construct with human primary chondrocytes, a synovial construct with human fibroblasts and human macrophages, and a neuron chamber to analyze the effect of each conditioning media over the neuron activity.
So for the podcast, can you kind of go through in a human, obviously neurons, the role of neurons is to send signals from the periphery to the brain. Can you just go through the roles of the cartilage and the synovium in a human, like an actual person? What are they doing?
Both of them are part of the joint. The cartilage, it is in between bones, so it helps to make a space and avoid the contact in between bones, and also contribute to generate a matrix and contribute to the synovial fluid to lubricate. We are talking about a healthy joint.
Yeah.
And in the case of the synovium, is the tissue that cover all the joint and also secrete a lot of trophic factors as well as a lot of compounds that helps to lubricate the joint.
Gotcha. And then so in osteoarthritis, do sort of all of these components start to break down, or like what kind of ends up happening? Yeah,
the dogma of osteoarthritis says that the cartilage, for any reason, traumatic or aging, starts to degrade. These products generate inflammation in the cartilage as well in the synovium, and that start a kind of degradation of the whole joint.
Okay. So now you've taken this and you're going to put it on a chip. Tell me how that works.
So we generate each of the constructs, and as I told you in the dogma, we started with generating cartilage degradation and inflammation through interleukin 1 beta, that is a common trophic factor used to modulate osteoarthritis. So to generate inflammation, generate cartilage degradation.
Okay.
So that's what we observe. And then we analyze the effect of these cartilage products on neuron activity.
Okay.
And what we observe is that in the presence of the conditioning media, so with the factors of the cartilage degradation, there is an increase in neuron activity, not only in the number of neurons active, but also in the number of spikes that they generate.
Okay, so that like damaged cartilage, the fluid from the damaged cartilage is making the neurons like fire more. So this potentially, like in a person, would lead to more like transmission of pain signals.
Yeah, exactly, and that is have seen in the advanced stages of the osteoarthritis, as in a normal cartilage, you don't have innervation.
Yeah. So there shouldn't be neurons there.
So at the very end, you will have neurons close to the cartilage degraded.
Gotcha. That's really cool.
So continuing with our model, as I told you, the dogma that cartilage inflamed generate inflammation of the synovium, we connected cartilage inflamed to synovial tissue, and what we observed was that there was an increase in the secretion of trophic factors, of pro-inflammatory factors by the synovial tissue. So we were able to mimic what is happening in an osteoarthritis joint.
Okay. So yeah, once again, that like damaged cartilage fluid is now we're looking at how it affects the surrounding tissue, and that surrounding tissue is releasing more like inflammation factors. So that's contributing to that environment of pain, swelling, discomfort, all of that. Okay. Gotcha.
And at the very end, what we did was we analyzed the effect of these cartilage, synovium, trophic factors, inflammatory factors over neuronal activity. We didn't observe any effect with this, but it's worth noting that we are applying trophic factors over neurites that are alone, so they are not innervating any tissue.
Right. So, they're not really connected to anything.
Yeah, in a normal joint, what you have is neurons innervating synovial tissue. So maybe this effect is masked because we just apply the trophic factors and we don't have the interconnection and the
neurons don't have anything to like connect with to talk to.
Yeah, so we are moving forward to a model where we will have the cartilage in contact with the synovium and neurons innervating the synovium, so to mimic what is happening in a real joint.
More of a full system, and then those neurons will have those other cells to talk to, and so maybe now you'll see the inflammatory factors might have a role, or maybe not.
Yeah.
We'll see.
Who knows? Yeah, but you also need some crosstalk in between synovial tissue and neurites. And as we are using a chemical model, we are trying to move to a mechanical stress model that is more close to the real problem for osteoarthritis.
Right. Yeah, and then if you can figure out like if any of these inflammatory molecules or something else that's in the fluid that comes from the cartilage, then that might become a target for like osteoarthritis treatment.
Yeah, the final goal of this project is to validate this model and start checking for potential treatments as well as treatments that didn't work in the past, or at least in mouse models.
Yeah, for sure. There's always like those steps, but that's really cool. And thank you for presenting this for the podcast.
Thank you so much.
Can you start by introducing yourself?
Oh, okay. My name is Lubya Salih. I am a fourth-year PhD student in Dr. Oban Mutal's lab at St. Louis University.
Cool. And so can you tell me what you've been working on?
Yeah, so my lab is interested in a subset of chronic pain, which is small fiber neuropathy or sensory neuronopathies. And those pain conditions specifically target the sensory neurons.
Okay.
And we are also interested in autoantibodies that are being produced in patients that have autoimmune conditions like HIV and Sjogren's.
Okay.
And previously in the literature found that patients with this autoantibody, 50% of them have chronic pain.
Oh, wow.
Specifically small fiber neuropathies or sensory neuronopathies. But it's used as a biomarker, and we don't know as a scientific community what they're actually doing, if they're actually causing the pain, or if it's a different mechanism. So we wanted to figure out what was going on with these autoantibodies.
Right. So like, is the immune system targeting the neurons, or?
We don't know. So we wanted to see if these autoantibodies that the body is producing on its own is actually doing or causing this pain.
Cool. Yeah.
Yeah, so the first thing that we were blessed to have patients that gave us their serum that were positive for anti-FGFR3 autoantibodies. And right now we have 4 patients, and their serum will be used throughout the project to induce this pain. So the first thing I did was I wanted to see if the FGFR3 would bind to the soma or to the DRG at all, to see if it was actually in fact targeting the DRG.
Okay.
So I used some IHC techniques to actually just see that. So these are rats, and then I stained with a secondary antibody after I exposed them to the autoantibodies. So that means it'll bind and it'll fluoresce. And we see that it's primarily in the soma of the DRG.
So the human antibody was binding the rat sensory neuron.
Well, it's just fluorescing.
Okay.
Because the autoantibodies are binding to the DRG, and then the secondary is binding and fluorescing.
Yeah, but the autoantibody came from a human.
Yeah.
That's wild. And so that's crazy.
And then I wanted to see if FGFR3 was even in the human DRG. So we got human DRG, which is really cool, from patients, and we stained them with the FGFR3 primary antibody. So this isn't coming from patients. And we saw that it did, again, bind to the soma primarily. So that was really interesting, and I wanted to see if these autoantibodies, what effect they were having on neuronal excitability, because that is a direct indicator of chronic pain or increased sensitivity to in vitro.
Yeah.
So I did some whole cell patch clamp over here, and what I did was I exposed the patient's, the DRG to the patient's serum.
Yeah.
So I did it for 30 minutes, and then I recorded the excitability as well as the rheobase, or the amount of current that is needed to induce an action potential. And I saw that there was a significant increase in the number of action potentials in the patient or in the DRG that were exposed to the patient's serum.
Okay.
And there was also a significant decrease in the amount of current that was required to cause an action potential. So these autoantibodies are causing an effect in vitro.
Right. So they're making these sensory neurons like more sensitive to stimuli. They're more likely to fire action potentials.
Yeah, and that's indicative of a pain state.
Okay.
Yeah. So the next thing I did was inject the serum from the patients into the paw of the rats, and we saw that they significantly increased the mechanical sensitivity of the rats using the von Frey method. And all of these individual patients are the red and yellows, and then the blue is the average of all the patients. So it's causing pain very quickly, just an hour afterwards.
And for a long time.
Yeah.
Yeah.
So we thought that was really cool, but we wanted to make sure that FGFR3 was actually the cause of this. So we did a Western. We took the DRG from the animals that had been injected with the autoantibodies, did a Western, and we looked at the pathway that FGFR3 is known to activate, which is the MAPK ERK pathway, and it's very prominent in chronic pain. So we saw that it was significant, the phosphorylated levels of the MAPK pathway were significantly increased. So that's confirming that the downstream pathway that FGFR3 activates has been phosphorylated, and there's an increase in the presence of those proteins.
Okay, yeah, so to explain this more sort of like in a layperson's terms, you were looking at the level of expression of the proteins in this pathway, and you saw that basically the activated version of the protein was upregulated in the DRG of mice that had been exposed. Rats. I use mice, and so I'm like, mice.
No.
But in the DRG of rats that had been exposed to the patient's
serum
with these autoantibodies.
Yes.
Inside of it. Cool.
And then the thing about these autoantibodies is that we only get like 200 to 300 microliters of them. So there's not a lot of them to go around to do a lot of experiments. So either we get more of them, which we get them from France.
Yeah, well, that's coming from far away.
Yeah. Or we created a autoimmune model using a plasmid that we injected into the shoulder of the rats and globally expressed FGFR3, which is that receptor. And then we looked at the mechanical sensitivity of the animals, which decreased over time after the injections of the plasmids.
Yeah.
And that was a great find for us because it's recapitulating the effects that we were seeing when we injected the autoantibodies.
So it's taking a lot longer though for these rats to show pain. Is that just because it takes a while for the virus to actually infect the target
So I'm not quite sure. That I found that was very interesting with the amount of time that it took. But I think, I mean, if we think about these patients, they are developing these autoantibodies over a period of time.
Right.
And we don't know when that started.
Right. So this gradual model might actually look a little bit more like the patient condition.
Yeah. And the autoantibodies, when we inject them, they might have some efficacy or better binding than our plasmid did.
Right.
So, yeah, that was cool. And then the next thing, because we know that the ERK-MAPK pathway can modulate sodium, voltage-gated sodium channels, we next use a NAV 1.8 and a NAV 1.7 blocker. There was no change when we used the NAV 1.8 blocker, which is the overshoot channel when you have an action potential. So there wasn't a change in the mechanical hypersensitivity in those animals, but there was when we used the NAV 1.7 blocker, Protox. Mm-hmm. So that is then going back to the flow that we believe these autoantibodies are acting through. So we think that the NAV 1.7, which is critical for keeping the threshold of the neurons, so that when it's activated by a stimulus, whether that be a painful stimulus, the neurons are firing more, which could go back to the neuronal excitability.
Okay, so I guess to summarize this part, because this is pretty technical, the autoantibodies are maybe acting on a pathway that affects channels and proteins that affect the sort of more baseline state of the neuron rather than like directly affecting how it fires action potentials.
Yes.
Okay, cool.
Yeah, so we're increasing the threshold.
Right.
Yes. So we saw that there was a significant change with the NAV 1.7 inhibitor.
Yes.
So in the future, we want to continue to look at these different patients, and we're using some epitope mapping to see where these other autoantibodies are binding on the receptors. So we can specifically parse out if there's a specific way that they're binding, which is causing this pain, and exactly what that's doing. And then I want to continue to look at the MAPK pathway, use some inhibitors of that. And then we're also doing a lot more EPHIS to figure out if the endogenous ligand of FGFR3 is causing, what kind of effects it's having in comparison to these autoantibodies. So we're trying to see how these guys are affecting these patients with autoimmune conditions and try to alleviate their pain.
That is so cool.
Oh,
thank you so much for walking through this for the podcast.
Of course.
And that'll do it for this episode of In Plain English. Once again, our presenters were Amy Nippert, Matias Preisegger, and Lubya Salih. You can find their posters on the website at inplainenglishpod.org, along with the previous episodes in this series. Our music is by Sam Brunwasser. You can find more of his work at soundcloud.com/visualsnowbeats. As always, you can download the paper and read the transcriptions at inplainenglishpod.org. And make sure to follow us on Twitter, Facebook, and Instagram at Plain English Sci. That's PlainEnglishSci. Thanks again for listening, and we'll see you next time for another episode of In Plain English.